Hi, everyone. Get a second countdown. Everyone, welcome to Night School. I'm Christina.
I'm Aria.
And we are a program of the California Academy of Sciences Nightlife program, which happens every Thursday night, which is why we're on Thursday night. Nightlife mixes together science, art, and culture, which we try to bring online to you. So tonight you're all here because you want to know more about snails. You may just think they're kind of like nondescript little brown things that you see on the sidewalk after it rains, but yes, they are that, but also so much more. So city snails, country snails, land snails, marine snails. We're going to talk about all of those tonight with our distinguished panel of gastropod loving guests. Who are they, Aria?
Yes, I will share them today. So in order of appearance, we first will have Dr. Mandë Holford from, she's a professor at the City University of New York and the co-founder of Killer Snails LLC, which is an ed tech company. And she's also a snail researcher extraordinaire and examines venoms, venomous animals. And today she'll be sharing about sea snails that fall into that category. Then we'll have Dr. Jann Vendetti, who is the Associate Curator of Malacology at the Natural History Museum, LA County. And she also directs a snail-focused community science project, which is very appropriately named SLIME, to kind of show us. And she'll be talking about, you know, the biodiversity that is literally right under our feet as we walk around every day. And finally, we're going to have Chrissy Piotrowski, who manages the Academy's very own invertebrate zoology and geology collection. And she's going to give us just a teeny tiny look into how many beautiful, unique, gorgeous snails in our scientific collections. And you will want to stay to the end of that because it is, there are some like wacky, gorgeous, amazing snails in there that just kind of blow your mind. So yeah, really amazing guests that we are excited to have with us here today.
And as always, tonight's stream is live. Anything could happen. So say hi. Thanks for letting us know what you're having for dinner. And yeah, feel free to drop any questions you have for our guest. Chrissy was actually pre-recorded, so you can drop questions. We can ask her. We can follow up with her. But Mandy and Jann are here live, and so they will happily answer your questions. So drop them in the chat. And without further ado, we have Dr. Mandë Holford.
Hi, everyone. Thank you so much. I'm so excited for this Snail Love Fest that's happening tonight. Thank you for the invite, Aria and Christina. So nice to be here in LA, but in New York, thanks to Zoom. So I'm going to share my screen and start the talk with my video, which hopefully you will see. Oops, you got the question ahead of time. Is this okay? I hope everyone's seeing the screen. Somebody respond. Yes. So today we're going to talk about the NightSchool Snail Love Fest that Aria described to you. I'm going to start off with sea snails, and then we'll go to city land snails, and then we'll go to collection snails. And so what I'm going to talk to you about are my very fave killer snails, which I think are agents of change and innovation. And you'll see why I think of them that way. So when you think of venomous animals, this is a hint, what do you think of? Snakes, scorpions, spiders, snails, all of the above, none of the above. Given that I told you these are killer snails, you're probably thinking snails. But most people, when they think of venomous animals, they think of snakes or scorpions or spiders. And they think of venom and venomous animals really as agents of fear, which makes sense because a lot of them are lethal and fatal to humans and can kill you. But when I think of them as agents of change and innovation. And the reason I think of them that way is because that's how nature has used venom throughout the tree of life. So we think that venom has occurred through convergent evolution. That means it evolved independently in all of these creatures. Everywhere that you see red is considered a venomous lineage. And so, yes, while scorpions, snakes, and spiders are the superstars and the ones you hear about all the time, most people don't think of sea urchins or platypus or the larvae of butterflies and moths or jellyfish and sea anemones or even starfish as venomous. And then, of course, snails. But we do think that 15 to 30% of all animal biodiversity is venomous. And so when most people think of snails, they think of them as this, the garden variety snail that you might see, like Aria was saying, when it rains, they come out. Or if you're a foodie, you think of them like this, on a dish all buttery and escargot and flavorful. But my snails are killer snails, and like I said, they're kind of on a mission. They're tattooed and leather up. They're pretty badass, and they're on a mission to feed or to protect themselves. And so this is what they do. Hidden under the sand is a cone snail. In orange, that thing is like a tongue. It's called a proboscis. On the tip, it has a radula, which is a false tooth that's filled with venom that it then injects into this unsuspecting fish, right? And so this is real time. We haven't sped it up. The snail will then rise up out of the sand and engulf the fish whole. So if this isn't innovation, I don't know what is, because here you have a snail that isn't even moving. I was going to say slow moving snail, but it's not even moving. And it's able to prey on this fish, which should be able to swim away. And you'll see this video again. So here the snail will, again, this is Conus bullatus. It will stick out its proboscis, inject the fish. Instantly, the fish is paralyzed. So the venom, it's very fast acting, it's very potent, and it's very specific for the targets that it's hitting. So this fish is very stiff. It's not going to move. But the snail didn't envenomate it with a high enough dosage of venom. And so you'll see that the fish will try to recover, but it's too late. I mean, it's gone. It's not getting out of that mouth. And so here's another version of how these snails eat. So this is Conus tulipa, and it is what we call a net hunter. And so it will engulf a school of fish, and then it will individually harpoon them all. So it uses its venom in the same way as the first one to feed, but it also uses its venom for defense as well. And so the killer snails that we study are mostly from the Conodeian superfamily, which comes in predominantly 3 flavors. So you have the cone snails, which are named that way because they look like an ice cream cone, and they produce conotoxins in their venom. Then you have terebrid snails, which are more sort of angular and also called auger snails, and they produce terotoxins, and turids, which produce terotoxins. In my lab, we focus mostly on the terebrid snails. A lot of the research on the snail venom was done on cones for the last 40 some odd years, and not very much was known about terebrids until we came around and started to work on their venom. To give you a little background on the snail anatomy, this is what it looks like inside the shell. You're peeling back the robe, sort of. So you have the venom bulb. The venom bulb pumps venom through the venom duct, delivers the venom into the radula sac. The radula are where we have those false teeth that we call harpoons. Get filled with the venom, and then it's delivered out through the proboscis and into the prey. The siphon is like a nose. It can sense when there's prey in the water. It smells, prey, and it's a chemosensory organ. And when there's prey in the water, that's when the proboscis will come out and the harpoon is ready to inject into the prey. And so this is sort of what it looks like as a schematic. And in real life, it kind of looks like wet spaghetti. So the bulb is looks kind of like a kidney. And then all of this curl and sort of wound up yellow stuff that you're staring at, that's the venom duct. And it can stretch out really long. And then the radulas are very thin, very hard to see without a microscope. And we call them harpoons because at the very tip of the radula, it's kind of shaped like a harpoon. And one of the functions we think it has is to tether the to the prey so that the snail is able to reel it in and without the prey getting away. And so all cone snails have this venom apparatus, but not all terebrid snails. In 1970, Miller identified 3 different hunting physiologies or anatomies for the terebrid snails. There's type 1, which doesn't have the radula sac, the venom bulb, or the proboscis. There's type 2, which has the venom bulb, radula sac, and the proboscis. And then there's a type 3, which has an accessory feeding organ and nothing else, essentially. And so the one we're most interested in was the type 2, because that's the one that's most similar to cone snails. And that's the one that we knew was actively using venom because it had the apparatus, the bulb, the radula sac, all those other things to deliver the venom. And so recently we've revised this anatomy, and we found beyond the 3 types that Miller described that there are actually 12 different types of terebrid foregut anatomy. And they're found all throughout the tree. And predominantly the venom gland is concentrated in certain clades that I'll show you in a minute. And what's interesting is that radula comes in very different forms. So the hypodermic needle form is the form that cone snails have. So we have that. But we also have these semi-rolled, flat duplex forms, which also are being used to deliver venom. And then there are other forms of the terebrid lineages which don't have the venom gland, but still are using and subduing their prey with venom. And so on its own, the terebrids represent all the various types of marginal teeth that you can find all throughout the neogastropods and all throughout the conodeians. And this is fascinating because this is a morphology type that's used. So when we go hunting for these snails, we're looking for the ones, it's kind of like hunting rabbits, right? We're looking for the ones that have the venom gland, the radula, the vaginal sac, and the proboscis. And so when we find them, we kind of get very lovey-dovey and we're very, very happy to have found our rabbits, so to speak. And so traditionally, the way you hunt these is you would go out to the beach and you collect as many of the same species as you can find. So size and quantity would guide what we collected and what we studied. When I started working on them, I wanted to learn from nature. So I wanted nature to be the guide to help us figure out which snails we would study, which venom arsenals we would investigate. And so by building this phylogenetic tree, we know how each snail is related to one another, kind of like how you're related to your family and siblings. And so we let evolution and diversity be our guide to let us understand more and to be able to ask more targeted questions about how the venom evolved in these species. And so we recently built a revised terebrid phylogeny in which we mapped the presence or absence of the venom apparatus onto the tree. And so the California clade, the terebrid clade, the Hostula clades, all are actively using venom to subdue their prey. And then we found these other clades, the McLauer, the Profunduit terebra, and the Puncto terebra clades are also more recently discovered to have active venom apparatus and actively using venom to feed their prey. And so venoms themselves are very complex mixtures. It's not one thing, but several things. So the venom is made up of small molecules, peptides, and proteins. In my lab, we mostly just study peptides, which means that it's a protein of less than 100 amino acids. And the amino acids are like the building blocks, kind of like DNA is the building blocks. Nucleic acids are the building blocks for DNA. Amino acids are the building blocks for peptides. And so each snail has anywhere more than actually 250 unique peptides in its venom arsenal. And the important thing to know is that no two snails have the same venom arsenal. And so whether you're comparing snails or you're comparing snakes to snails, venoms are very species specific. And so each cocktail is quite unique. But what's interesting is even though the cocktails are unique, they kind of are acting like a cluster bomb. In my case, we're focusing mostly on the peptides in this cluster bomb. It's not the big bomb, but all the little ones that do the damage, right? So in this cluster bomb, all of these peptides, whether it's from snakes, scorpion, spider, snakes, they're all targeting very important things, and they're all addressing cellular physiology, whether it's at the blood level, so they're hemotoxic, at the brain level, they're neurotoxic, and at the membrane level, they're cytotoxic. They can also be myotoxic for muscles and necrotic, where they cause, death to cells. But these are the big major things. Basically, what the venom is doing is it's disrupting normal physiological function. So this cluster bomb invades the prey, invades your system, is basically trying to shut down how you would normally work. And the best part is it's very good at what it does. So venom is a very successful molecular innovation. And scientists are excited to study venom in snails and in snakes and in other organisms because we know that it works, right? You just saw a snail eat a fish. And what it's very good at is manipulating cellular communication. And so what we're studying is how these venom peptides can be used to manipulate signals, in particular malfunctioning signals, to learn more about how these channels work on the surface of cells and to develop compounds that could be useful for diseases and disorders. And so the killer snails are found in tropical waters all around the world. So everywhere you see a little pin drop is where we've been on exploration to identify these terebrid snails. We do this in collaboration with the Philippe Boucher's group in the Paris Museum of Natural History. And so there are over 400 described terebrid snails. So far, we have about almost 200 of them on our tree. So we're almost at 50%, which is really exciting because we want to be able to fully be able to reconstruct the evolution of venom in this group. And to do that, you need to collect as many of the species as possible. And so these snails are found in tropical waters all over the world, and they're also lethal to humans, certain species. We should not be worried, though. Most of the fatality occurs along the Indo-Pacific, where you have the highest biodiversity of these snails. Everything in Australia will kill you. You know that. So stay away if you can. In New York, I'm presenting from New York. I'm very safe. We don't have the tropical waters. California, Northern California is fine. Southern California, in Baja area, you do have certain varieties of the snails, certain species, which can be fatal. So be careful if you're out in Baja and you're doing, fun stuff in the water. But mostly they're nocturnal. If you see a pretty shell, just don't touch it. It's the first thing they teach you when you learn to scuba dive. If it's pretty, don't touch it. If it's ugly, don't touch it. Just don't touch it. Just dive. And so, because these things are fatal to humans, when people think, how are you taking venoms and using them for drug development, which is what we are trying to do, it's like, we have evil Captain Venom. Captain Venom will kill you. It's lethal. Yes, if we were to inject all of a venom arsenal into a person, it would potentially kill them if it's from the species that's fatal. But as Dr. Envenema, what we are doing is trying to identify specific peptides in the venom and understand how those peptides are able to manipulate signals in order to alleviate malfunctioning signals like pain or cancer. And so all of these animals, the Gila monster, the Chinese centipede, Brazilian land snake, and cone snails, all of them have been various stages of development for drug discovery. Some of them are on the market already. From the Gila monster, we have Bieta, which is being used to treat diabetes. From the lance head snake, we have captopril, which is used to treat hypertension. And from our own very special cone snails, the first drug from the cone snails that's on the market is called Prialt. It's used to treat chronic pain in HIV and cancer patients. So we know that we can use specific peptides from the venom of these animals to develop drugs that can make it to market and actually help to alleviate human diseases and disorders. And so in my lab, the cone snail drug, Prialt, that's used to treat pain, it has one major side effect in that it is not peripherally active, and you have to get a spinal tap to deliver it. So its distribution, while it's much more effective than morphine because it's not addictive, it doesn't lose its potency over time, it does have a serious drawback that you can't just administer it through the bloodstream or pop a pill orally. So one of the major things that we're doing in my lab is trying to figure out how we can identify new peptides from terebrate snails that are peripherally active and can serve as analgesic compounds. And we're doing this in collaboration with Adam Clatteridge Chang's group in Singapore, in which we are using whole animal fly models to look at phenotypic assays in which we either feed the fly the peptide, the terebrate peptide, or we inject it peripherally, and we look to see if the fly can tolerate heat, basically, as a sense of analgesic pain. The other area we're looking to use these peptides is in cancer, where we treat cancer kind of as a channelopathy. So channels are these gates on the surface of cells that allow things to move inside and outside of the cell. And so we want to use the specificity of the peptides to target tumors in which certain channels are being overexpressed. So in purple is the channel, and in blue is the purple is the tumor, blue is the channel. And then we look for specific peptides that will target those overexpressed channels that are specifically found on tumor cells and not on the healthy green cells. So minimizing the effects and maximizing the selectivity for the drug, minimizing the treatment. Another area that we're starting to get into is looking at what we call vampire snails. And so this is in collaboration with Maria Victoria Modica, who's in Italy. And she's identified certain compounds within these vampire snails that are very good at, acting as blood thinners or anticoagulants and other kinds of anesthetics. So this is called Colubraria. It sends its proboscis under the gill of a sleeping fish, and then it's actively sucking the blood of the fish. They do not kill them. That's why we call them vampire snails. They basically just suck the blood out of them. And so that's very good for a lot of these things that can potentially be useful for human diseases and disorders. And so I've painted a very rosy picture, but I want to leave you now with a little bit of reality about the history of peptide drugs. The first peptide drug was discovered about 100 years ago, and it was insulin, and we used it mostly by injecting, and we still use insulin by injecting. Then Bruce Merrifield, who in 1963 developed a technique called solid phase peptide synthesis, which allowed us to make synthetic versions of these peptides, and it really revolutionized the field. And so similarly, we're hoping this approach that we're using, the venomics approach, which combines lessons from nature with the rise of omic techniques, transcriptomics, proteomics, metabolomics, genomics, to help us identify the peptides in these snails very quickly, and then help us to characterize their function towards drug discovery. But it's a very bumpy road, right? So in 100 years, just think, we've had insulin, and we still pretty much use it the same way that it was discovered, in that it has to be injected to be most effective for most people. And so these killer snails really are on a feeding frenzy. So in A, you see a snail that basically will drill a hole into a bivalve and then suck the animal out. In B, there's a snail that uses its shell like a wrench to pry the bivalve open, and then it will have itself a tasty meal. I showed you C, which are the vampire snails that suck the blood out of fish. In D, you have these snails that feed on corals. And in E, those are my very special killer snails feeding on fish, worms, and other snails. And so this girl from Brooklyn gets to travel the world to look at killer snails. And so I have a little bit of swagger because it's a lot of fun. And we go out. These snails are mostly nocturnal, so you get the most of your best catch when you go out night diving on the snail. So this is us on one of the last exhibitions we did before the shutdown in Papua New Guinea. We go out at night. Sometimes it's raining, but you still go out because you must collect. And so we come back, everybody's smiling because we have different things. This trip in particular was funded by the Monaco Foundation. And so that's the Prince of Monaco. So sometimes you meet a celebrity in the field. He gave us money and he came to check to see what his money was doing. So we showed him exactly what we were doing in the field. And mostly it's a good time, right? You're out meeting new people, learning about different cultures, and exploring marine biodiversity. And so I hope that I've shown you that venom and venomous animals, especially venomous snails, aren't just agents of fear, but they're actually agents of change and innovation. And this could be you on our next exploration, right? You can get all, tattooed and leather up and out for the kill with the killer snails. And so with that, I'd like to thank all the lab members and of the funding agency that support their work in our lab. And thank you, and happy Snail Love Day, everyone.
Oh my gosh. Hi, snail fan here. That was amazing. Thank you so much for walking us through your work. And we have many questions coming in, so I'm just gonna get right into it. Okay. So first, Deanna asks, what are the predators for these cone snails that you showed? Do they have predators?
They do. Their predators are things that are hard-bodied, so like lobsters that can crack the shell, but for whom they can't penetrate and inject them with their venom because the harpoons don't work on those hard lobster shells. Yeah.
Gotcha.
So lobsters and crabs are their major predators. Yeah.
Yeah. And next question, how do these snails actually sense their prey? Like, do they rely on any kind of eyesight or
No. They have eyes, but they really don't use them because they're mostly buried in the sand for the most part. So that they have this chemosensory organ, the siphon, that sticks up out of the water. It kind of looks like a worm maybe to a fish. And they can smell what's in the water based on those chemosensory organ and odors, and then they're able to then put out their proboscis when they sense or smell that there's a prey in the water.
Gotcha. Wow, that's amazing. And you said that it maybe looks like a worm to a fish, and does that attract their prey a little bit?
Fish to come closer, I think, and then the fish doesn't realize what it's getting into because most of the snails stay buried under the sand.
Right.
Or whatever substrate they're in.
Yeah. Oh my gosh. So cool. And Derek asked, how much of these snails' mass is actually taken up by that venom bulb? Because it looks pretty big in there. That like
Yeah,
organ.
the bulb is actually very big. It really does look like a kidney bean. Um, but a lot of their body is very tightly coiled up within the shell. So the venom gland itself can go almost the length of the snail and longer. And it's the bulb, while it looks very big, it's not that big in relation to the rest, like the foot. The foot is the biggest organ of the snail.
Gotcha. Okay. All these like snail anatomy questions. It's so cool to get to know them better. And Heather asks, do you have any kinds of special protocol for collecting and studying snails that can be fatal to humans?
Oh yes, we do it very carefully. We use high-tech salad tongs so that you can pick up the snail and drop them into your bucket or something. We also use scuba gloves, and again, they're snails, right? So they're not like snakes. So snakes are very aggressive. Snails, when you pick them up, they will recoil into the shell. All the envenomations that happen for the most part or predominantly either scuba divers who see a pretty shell and they pick them up and they put them in their wetsuit. And if the shells feel like constrained, that's when they'll put out their proboscis and try to envenomate you. But for the most part, if you pick up a shell, it'll recoil, close its operculum, which is like the door to the shell, and stay in there. Doesn't want to be bothered. So if you're very quick in picking them up and putting them in the bucket, you're safe.
With high-tech salad tongues.
Yeah, with high-tech salad
with
tongues.
high-tech salad tongues. Oh, good. And Danny is asking, what's like the method of transportation for these snails from the natural environment to the lab?
Oh, we usually, if you have to collect, we usually dissect in the field, and you take the venom gland, or you will actually take the RNA or DNA extract in the field and take it with you. But if you have to take an animal home or ship one home, you have to. They can hibernate, kind of like bears, so they know how to lower their metabolism. And so you basically will keep them in a tank and then slowly decrease the amount of oxygen that's in the tank until you're ready to travel with them. And they will then hibernate for the most part until you get them back into another tank and then increase the oxygen again for them to be able to survive the plane trip.
Oh my gosh. Wow, snails hibernate. That's a fact I did not think I would hear today. That's amazing. So cool. And okay, so shifting gears a little bit, question from Ria. As you're studying the evolutionary history of these animals, can you see how the various peptides evolve through history and how they relate to each animal's niche?
Oh yeah, that's a very good question. Some of the work that's being done in Toto Oliveira's lab, where I did my postdoc, he's been able to identify that certain snails are feeding on specific prey. So their venom arsenal is quite specific to their prey. They found in cone snails, depending on if they feed on fish, worms, or other snails, they have a venom arsenal, meaning the peptides in their venom are quite specific for that prey. We haven't found that with our terebrids, and I think it's because unlike cone snails, all cone snails have a venom apparatus, but not all terebrids do. And so the snails are hunting in a diversity of ways, and so their arsenals are being used quite differently. And mostly terebrids also feed on worms. They don't feed on, or it hasn't been recorded that they feed on fish and other mollusks. So their meal is quite strict compared to cone snails. But you can try to trace their peptides to specific predation organisms.
Amazing. Cool. Well, my final question for you in the spirit of the Snail Love Fest, what was the moment that you fell in love with snails?
Oh, I fell in love with snails when I saw that video, that crazy video of
Yeah, I hear that.
of a snail eating a fish. I had no idea. And then I was like, really? And then I learned it was peptides, and that's where I did my my PhD at Rockefeller. It was all in peptide chemistry. And so I was sort of hooked. I was looking for a way to bridge my love of doing biomedical research with my love of natural histories and going to natural history museums. And I wanted to find a way to bridge those two things. And so venom allowed that to happen, right? Because venom is, it occurs in natural organisms like snakes, snails, and spiders, but it also is very much biochemical compounds that make up the venom. So it was a way to sort of bridge what I knew with what I loved, my heart, my mind, I like to say, to pursue something. But seeing a fish eat a snail is just crazy. And then when you see them eat worms and other snails too, should have shown some of those videos too. That's like nonsense. So nature's beautiful. And it has all the
nature is beautiful.
We just have to figure out what it is and how it works, right? And we could probably live forever.
Oh, seriously. Oh, that's incredible. I'll send everybody go do some searching. All from the snails. And thank you so much, Mandë. We really, really loved hearing about your work and having you here. And
thank you for the invitation. It's been so nice.
Yeah, and we're going to go ahead and bring up Jann to talk about land snails next.
Hi. Hi, everybody. Can you hear me okay? I hope so. All right. Mandë, that was amazing. And yeah, anytime you want to come to Los Angeles and visit my collection, you are welcome to. And I'm a big fan of natural history collections. That's my job. I'm the associate curator of Malacology, or mollusks. The study of mollusks is Malacology, at the Natural History Museum of Los Angeles County. And I'm going to talk with you all tonight about land snails. And one of the projects that we have at the museum to document the biodiversity in land snails, and the incredible biodiversity that there is around you and me and anybody on Earth. So a shifting gears from marine snails. And one thing I wanted to start with here is this is a picture of Otala lactea. The snail is, it's called the Spanish milk snail. This is in Los Angeles, and it is estivating. And estivating, Mandë discussed in one of the questions, she said that snails have sort of a hibernation strategy. And in land snails, that hibernation strategy is called estivation. And they can do this for months. So I'm not going to talk about that very much, but I just wanted to bring it up. This is what my field site looks like. It's a collection. I don't do nearly as much field work as somebody who is studying behavior or biochemicals from organisms. I am building our collection and tasked as a curator with studying the biodiversity within our collection. So this that was just a picture of what one of the aisles of our cases of specimens. I tell people it should look like a well-organized closet, but it's not an exhibit, right? It's just it's those specimens housed so that they can be used by scientists. And the way that I usually relay this message is that we think of our collection, and anybody at a museum of organisms thinks of their collection as a library of biodiversity. So I say, instead of thinking about a library of books, think about this museum collection as, instead of books, you have specimens, and those specimens are cared for. Those specimens are loaned, they're returned, they're kept in an organized fashion. There's a database. Sometimes in ideal situations, that database is online. So anybody who wants to know about where to find those specimens can, and they can use the information from those specimens to do good science. To give you a handle on the mollusks, so this is just a picture of some North American seashells. Mollusks are often called seashells. There are about 100,000 species of mollusks, and most of those are gastropods. Most of those are snails. So this is just showing you the gastropods among their friends, the other mollusks. So you can see cuttlefish and a nautilus, and an octopus. Those are cephalopods. A chitin is a chitin, or a Polyprocophora. And then over to the right are the mussels, and their friends are the oysters and other bivalved things. Those are the bivalvia. The gastropods are extremely diverse. You got a sense of this from Mandë's talk. And one of the largest innovations, and I would argue one of the biggest innovations in all of the animals, was the transition from sea snails to land snails. And that's quite a big deal. And there's almost as many land snails as there are sea snails, and many yet to be discovered. So you may be asking, why do I have birds and mammals? And this is to give you, on the slide, this is to give you a handle of, so imagine now we have that 100,000 species of mollusk. Of that, there's about 40,000 species of just land snails. So just keep that in mind. 40,000 species of just land living snails. And as a comparison, there are, let's say, about 15,000 species of birds. They just increased it. It was like 10,000, and then some people say it's 19,000. So let's split the difference and say it's around 15,000. 15,000 species of birds. Every extant living bird, 15,000. And below that, the mammals there. And so think about every mammal that you know, a blue whale, a wolf, coyote, hamster. There's about 5,000. Every single, and a quarter of those are bats. So think about all of that biodiversity in mammals, a tremendous amount of biodiversity in birds, and that land snails have more than twice that. So I became interested in terrestrial or land-living snails and wanted to learn more about them. We decided to use a citizen or community science approach to do this, and also to engage the public. The program the project that I came up with is called SLIME. That's my colleague who made it into an acronym. It stands for Snails and Slugs Living in Metropolitan Environments, SLIME. He's a very clever entomologist. My idea was Snail Project. So he came up with a much better name than I did. This is a contributory project. It means that people who post their observations to the SLIME project on iNaturalist, at least right now, have to do it purposefully. So it doesn't aggregate if you know iNaturalist, which many of you probably do, it doesn't aggregate your submissions, your observations automatically. You have to add them, and I may change that. But anyway, in the last 5 years or so, we have just under 19,000 observations and around 130 species. This has really been a remarkable project, and it's taught us a lot about the biodiversity in Southern California. So the SLIME project is the 10 counties of Southern California. It's taught us a tremendous amount about the biodiversity of this group. We also have a number of events. One is called Snail Blitz. Snail Blitz per year, once a year, is about a 2-month project, and we try to really promote it a lot on social media to get as many observations as possible. And some of our most interesting sort of discoveries about where snails are and what snail species are in Southern California are from Snail Blitz each year. This has also been the inspiration for an Atlanta project called Atlanta Slime that a colleague of mine started. And then the Burke Museum, our neighbor up north, the Burke Museum in Seattle, also began a slime project called Slime Pacific Northwest. And you can see that they have 37,000 observations, and that's because they're in the Pacific Northwest. I'm so excited for them to do this. I do feel a little bit hurt that their numbers are so much larger than ours. But as you know, in California, especially Southern California, we're dealing with quite a profound drought, and Pacific Northwest doesn't have that issue, and they've got lots of snail biodiversity. So in the time I've got tonight, I want to leave you with a kind of a big idea. And this big idea is that because I'm studying snails in mostly urbanized area in greater Los Angeles and the developed part of Southern California, that I want you to think about urbanization and its influence on biodiversity as a filter and a homogenizer. And this is not an idea that's unique to me. People have been talking about this idea for quite a long time. And this is, remember, just urbanization as a filter. And what I mean by that is if you have a species that would live in a particular environment, but urbanization has changed that habitat, now you have likely filtered out that particular species. And remember, and you all know this, that if you plant a pollinator garden, even in like an apartment window box, you can get a great diversity of native flying pollinators, bees, wasps, flies, things like that. And that can be a sort of a haven for those species. That's not the case for land snails, right? They're not flying. They are limited to the habitat where they evolved generally, and they don't move around very much. So you can build a habitat for them, but they're not going to get there, right? I tell people that they're not going to cross the 110 freeway to get to the museum's beautiful nature gardens. They're stuck where they are. So anyway, this means that these species that would otherwise be in a habitat are pushed out because of urbanization. So one of these is Helmintha glyptotrascae. This is almost an LA endemic species, native species. You have them, we have them all throughout California. This genus Helmintha glypta, it's a mouthful, but Helmintha glypta and Helmintha glyptids, they're friends of many other species, are all over California, and they are very understudied. So some colleagues and I are trying to work that out. Here are some examples of, this is called the Trask shoulder band, Helmintha glyptotrascae on iNaturalist. These were observations made by contributors to the Slime Project. And just to kind of get a sense of how difficult it is for snails to survive urbanization and habitat change, I'll just read this quote for you. There are more land snails listed as threatened, endangered, or extinct by the Union for the Conservation of Nature than birds, mammals, reptiles, amphibians, and fish put together. So this is a picture of, you can see, right, this is an iNaturalist observation that I made in 2017. This was while I was walking back from teaching at a community college in Los Angeles County, and this was a tiny little plot of dirt. You can see this right next to an elevator and a staircase. That particular plot of dirt has now been paved. So, and that's a native snail living there. And this is just like a beautiful, sad, but beautiful example of what I'm talking about, that urbanization and development can very quickly change the habitat of these native taxa. And here's another one. This is just in March. This was on a different side of campus where there was some relic habitat left over, and these snails are coming down from the surrounding untouched hillsides and just eking out a living right around campus. But you wouldn't find them anywhere else in the urbanized part of Los Angeles. This is what some of these specimens look like in our collections. This is how we house them, right? They're shells. They have data that go back for, in some cases, more than 100 years. Just as one example, this is what one of our specimen labels would look like. This specimen is another Helmintha glyptotrascae, and it was collected in 1936 from Hermosa Beach. And this, so I'll go back for a second. This was collected in 1936. As of the 1950s, this, you can see that red teardrop shape, that was where it was collected, 24th Street in Hermosa Beach. And as of the 1950s, this is what that habitat looked like. It's all houses now. So now there are no native land snails found in this habitat. But instead, if you look on iNaturalist, the most common snails, found in Hermosa Beach in this particular neighborhood are all introduced. Garden snails, Ambigo limax slugs, the milky slug, and the species on the very right called Oxychilus, all introduced. So this is, I just want you to think about now that this is facilitating urbanization. In addition to being a filter of some species, is now also facilitating the introduction of many other species. So these snails are coming in with people's plantings from Osh and other garden centers, the Home Depot and whatnot, and they're coming from around the world, these different species, as hangers-on to usually planted landscaping plants. Here's another native species. This is Glyptostoma gabrielense. This is native to the San Gabriel Mountains that are right next to the city of Los Angeles. It's about the size of a quarter. Its body is blue. It's a beautiful snail. Almost nothing is known about the natural history of this snail. It has one sister species that's found in San Diego County. So these are the only two of the group that it's in. It's sort of, it's not exactly a living fossil, but it's certainly from a depauperate lineage. And almost nothing is known about this, and it is not found in urban environments anywhere. So it's only found in native habitats. Okay, so just shift to the other point I wanted to make is urbanization as a homogenizer. So it's a filter, right? It's filtering out many taxa, and it's only allowing certain species to live in these urban environments. But then it's also, because it is facilitating the introduction of a lot of different species, it is also, the force of urbanization is creating homogeneous or similar faunas, what we would call malacofaunas, snails, different snails all throughout the world that would not be that way naturally. So for example, this is a picture. This is a contributor to iNaturalist. This is from southern Spain. And this photo could have been taken in Los Angeles. All of these species, in yellow is a species called Cornu aspersum, the common European garden snail. In pink is Otella lactea, the Spanish milk snail. And in white is Theba paisana, or the Italian snail, white Italian snail. And I'll just say this point again. This photo was taken in southern Spain, but all 3 species are found in abundance in Southern California because they were introduced. So our snail fauna in Southern California, and you all have these species in Northern California too. Our species in California are very similar. Our introduced species are similar to native faunas elsewhere in the world. As an example of this, when I also study slugs, which are snails without shells, like lineages of snails without shells. And I use this book called Slugs of Britain and Ireland. Why in the world would I use a book of slugs of Britain and Ireland? It's because our slugs are overwhelmingly slugs from Britain and Ireland because they were introduced here. And so this idea of biotic homogenization is across the board with a lot of different taxa. Because of globalization, so many different species of many different lineages are being introduced around the world. So you get this phenomenon. It's fascinating. I mean, it's an experiment in real time that has a lot of fascinating implications. This is a European species that we found through the Slime Project. So this was not discovered by me, but I was really happy to co-author a paper with some members of the community who did find this snail. This is called Xerotricha conspurcata. It's European. This is another one, Prupioides albilabris. This is a southern U.S. species that was found in Southern California, introduced by anthropogenic means, by people somehow. Cochlocella barbara, the same. This is a pest snail throughout the world. We have it in Southern California. You all, I think, have it. Maybe you don't in Northern California, but in San Diego, they also have it. This is how tiny it is. This is where it's found, in little cracks and crevices. If you see something like this and you use iNaturalist, or if you don't, please use iNaturalist and make an observation, because what you might think of as an insignificant little snail could be the first record of that species found in wherever you are. And somebody on iNaturalist will find it, and it's really quite remarkable to be able to work collaboratively with members of the public. This is our friend Otella lactea, estivating, or in snail torpor or snail hibernation in Northern California and Southern California. Here's a whole bunch of iNaturalist observations. Because of iNaturalist, my colleagues and I did a study on that species and found that it had at least 2 different introduction events. Genetically, we looked at that. We did the same thing for this Italian snail, white Italian snail. It's found in San Diego County and San Pedro, and elsewhere in Los Angeles County. And just doing some simple genetics, we found that the Los Angeles County the Los Angeles County population was most similar to snails of the same species from the island of Malta in the Mediterranean. And in San Diego County, they were most similar to these snails from Morocco. So we also have multiple introductions of this introduced snail throughout Southern California. This is something that's gone on in all throughout the world for a long time. This was in parts of Los Angeles. I just wanted to show you. This was from 1935, and this is a person attempting to wipe out the pest horde of white Italian snails by whipping flaming torches over the infested area. So our habitats in Southern California can become infested with these snails because they have very few natural predators, which is another interesting phenomenon on its own. I don't think anybody's going to be whipping flaming torches in Southern California right now, but they did, because these snails got so out of control. This is Cornu aspersum. I just want to show you a picture. This is introduced. It's an escargot snail. This is Rumina decollata. The snail was introduced to eat this snail as a not necessarily a great idea, but this snail is now all over Southern California and elsewhere. And I'll stop there so I can answer your questions, but feel free to contact me if you have any. Thank you so much.
Thank you, Jann. That was you were going so fast at the end. But we do.
Sorry about that.
No, it's great. Yeah, because we do have a lot of questions about these.
Let's do it.
Snails. So first one's kind of selfish, which is, Are there any plans for a snail blitz for Northern Californians?
Oh my gosh, let's do something, Cal Academy.
Okay.
Sure.
Okay.
Let's do something.
Okay. Yeah, we know some people who, use iNaturalist a lot. So.
Yeah, I think you all know about that, don't you?
Yeah.
You know, National Geographic, you know something about that. That's right.
So actually, Mandy had a question.
Oh my goodness.
Yes. And she asks, how are the snails introduced to Southern California? Is it predominantly people bringing pets or for agricultural reasons related to crops?
They're bringing them accidentally, if I understand your question. Yes, it's accident. It's by accident. Yeah. They are hitchhiking onto potted plants and in soils and in if you imagine snails lay eggs. They're tiny little, like if you made, I don't know, how big is it? You could probably fit like at least a dozen on your pinky nail. Oh, they're tiny. And if those are mixed into, if you don't see a snail or a slug on a potted plant, it doesn't mean that there aren't snail eggs in it.
Right.
So it's mostly by accident. However, there are the Spanish milk snail and the white Italian snail are both eaten in North Africa and parts of Southern Europe. And it is possible that there were mostly Southern European, like Italian immigrants in parts of San Diego and parts of Los Angeles who brought those snails with them. And there's documented accounts of this. But there's also a how should I say this? There's people blaming, like, oh, the immigrants did it. Those Italian immigrants brought in these snails, when in some cases they probably did not. But in some cases it's possible that they did. So large edible snails could have been introduced by people, but they've been introduced multiple times. And I'm convinced that some of those events were accidental. And if possible, I mean, it is possible some were on purpose.
So outside of, I remember the example of one snail brought over to eat the other snails. Have there been other instances? If somebody brought snails on purpose, would it be for that reason usually, or are there other reasons you would?
Some for biological control. I mean, that was how it was that snail that looked like the top of its shell was cut off. It's called Rumina decolata. That snail was brought over on purpose to eat the eggs of the common European garden snail, which was a citrus pest. So it was in the sort of heyday of Southern California's citrus groves, and that was a pretty profound pest snail. So they let loose all of these predatory snails to eat the eggs of Cornelius bursum to save the citrus crop. But those snails, those little decolate snails, they don't know where their boundaries are. So if they get up into habitat of native snails, they'll eat their eggs as well. And so that's biological control can be a really tricky situation and with a lot of unintended consequences. So those were introduced on purpose for, as far as I know, in Southern California, no other species was introduced no other snail species was introduced as a biological control. But as an escargot or eating snail, probably. Yeah.
Here's a question from Raya. Are snails we find in our San Francisco backyards completely trapped on our block? And do the roads keep them from moving elsewhere?
That was a good question. I think that when it rains, they'll cross the street. I think they'll move. I mean, how much a snail moves in some period of time, I don't know. It depends, right? Not too far, but within a neighborhood, I think it depends on how humid it is and how much it rains, because they'll move around. Like they will do nothing. They'll estivate for sometimes quite a while. And then when it rains or the humidity is high, they'll sort of wake up and they'll go do everything that they need to do. They'll mate, they'll find each other, they'll mate, they'll eat. And then once it dries up again, they'll find a place to hang out and they'll crawl up a sign or the side of a building or a leaf of a plant, and they'll just hunker down until the conditions become favorable again to go out and do all their snail stuff. So, they can I don't know, they can cross a street. Yeah, they wouldn't be trapped to your block. Yeah, there are people who study the range, how the range of a certain introduced species changes, and that's one of the things that is really great about iNaturalist is we can, if we know where the boundaries are of a population, we can use iNaturalist observations to track over time how that population is expanding, or where and where it's going, and over time how long that has taken. And that's really useful information for pest species.
Yeah, great. Question from Derek. How do you grow the museum's collection, and what are the best practices with regards to more endangered species?
Oh, such a good question. So with introduced taxa, we had a very small, at LA County Museum, we had a very small collection of native and introduced land snails. And the only, local species. And I think it's because there was this idea that, and this is a problem that museums used to have and sometimes maybe still do, that the exotic was more interesting than the local, and that curators went elsewhere and collected and then brought those things back. And instead of that model, we're focused much more on studying what is local, and it's been ignored by a lot of museum scientists for quite a long time. So there's a lot of work to do. So anyway, in collecting, I usually collect what I feel like I need for a study. If it's genetic, and I'll collect a dozen or so snails, and then the live specimens we sacrifice and we keep in ethanol so that we can use their tissues for dissection and for DNA. And then if something is endangered or thought to be endangered, most snail species have not been evaluated for an endangered status. But if we suspect that they are a small population, you can get DNA using like a cotton swab from their foot slime, and it's not like 100% of the time, but maybe 50% of the time.
Interesting.
It's sort of like, if you do a buccal swab or a spit to get your DNA sent off to Ancestry.com or 23andMe or whatever. We do that with these little snails on their slimy foot, and then if we do a DNA extraction of that slime, we can often get enough cells of their body to get DNA. So in that sense, we don't have to sacrifice as many animals. However, we do feel like it's part of our job to be a repository of specimens so that we have an actual physical record. But that's also where iNaturalist comes in, that we can know information about the living animal and where it is without having to add it to the collection.
Yeah. Great, thank you. I also just love the idea of like a snail 23andMe, and all these snails are like, oh my gosh, my family is so big.
Well, that's what Mandy studies, right? Mandy's figuring out who all the long lost cousins and all those. Yeah, absolutely.
Get a phone call one day. Yeah. Anyway, so what kind of benefits does having a variety of snails have on the environment? Like, why is it good to have lots of different species?
For the environment, I mean, they all play some role, right? They all are eating something. Often snails, not always, but often they're specialists. So they are sometimes eating something very specific. They're the prey item of something very specific. So there's imagine in high school, like you learned about food webs, right? It's not just a chain, like a food chain. It's more of a web where there's so many interactions that happen between any particular species and other taxa in its environment. So if there is only one native slug in, well, yeah, that's probably true. In Los Angeles County, one. Yeah. And there's nothing that's inherently, it's called the Hempfield's Western slug. And it's this cute little silvery slug I've seen a couple times, but that's it, right? And there's nothing inherently wrong with Southern California, chaparral or oak woodland environments that there's only one slug versus somewhere else where they would have many. There's nothing better or worse about that. It's just that's what evolved, and that's what's there, and that's what's allowed. That's what has survived given climate change of many millions of years.
Mm-hmm.
So, but yeah, they're all of these species, like anything, are these remarkable survivors in time of and they have adaptations for their specific environment. And habitat change does a lot very quickly to wipe them out, and they can't get up and move and relocate. So so they're often a harbinger, like a canary in the coal mine of habitat change, because they'll be the first to go, and you can't attract them back.
Oh,
they're not flying in, right? They're there. They're not. So they're they're also, native snails are also an indicator of sometimes what's called a relic habitat. So a habitat that hasn't been messed around with very much in a long time. So if you have a native land snail, it can be sometimes considered an umbrella species, a species that if you save that species, or if you make habitat available that is untouched for that species, it'll protect many, many other species in that environment.
Great, thank you. Okay, and we have to ask you, what made you fall in love with snails?
I grew up on the East Coast, and I spent some time in Maine in the summer visiting my grandparents, and I got hooked on intertidal organisms. I just couldn't. It was like the most magical experience, and I felt like that always, ever since I was a little kid. And then when I came to this museum, I was like, well, I'll study my marine snails. And then it turned out nobody knew anything about these land snails. So I became, I sort of converted. I converted to land snails from marine snails. But they're wonderful. And I think of looking in leaf litter, like looking in a leaf, like a patch of leaf litter to me is like looking in a tide pool.
Oh.
Like it's similarly, like you pick up the seaweed and you look underneath and you wait and you see what crawls. And that's the same thing. If you lift up that leaf litter and wait, I mean, the biodiversity is just stunning, absolutely stunning. And it's as inspiring as a tide pool is. So these little snails have just won me over.
Oh, so do you love one more or the other now, or do you love
them like my
equally?
children? No, I can't. No, they're equal. I love them equally
than the other.
I love them equally. I mean, I've converted to paying more attention to the land snails, but that was because they were neglected. They were neglected for so long.
Yeah.
So now I'm giving them their what they're due.
I'm really glad you are. I'm really glad you are. Thank you so much for joining us tonight, Jan.
This was great.
This was awesome. It's so fun to have a snail love fest. Okay. Well, now we have our tour with Chrissy, and you can look into our collections and what they look like. Yeah, and thanks again, Jan.
Thank you.
Bye.
Hi, welcome to the Invertebrate Zoology Research Collections. You're in our dry collection room, which is one of several rooms that we have. We also have a fluid preserved collection rooms upstairs, and we have larger specimens that we store in a tank room on the other side of the building. Right now, we're going to look in our dry collections and see some of the huge collections of snails that we have. We have I want to say in the mollusks, which are the snails and the bivalves and related groups, cephalopods, we have about 300,000 containers of mollusks, and so that translates into many millions of actual shells. So our shell collections don't have tissue for the most part associated with them, so that's why we can store them dry. If there are tissues associated with them, then we store them in ethanol, and those are upstairs. So come on down the aisle. Here at the Academy, we have a very large marine snail collection, and we also have really historically important and pretty large land snail collections, so terrestrial snails, and also freshwater collections. So snails are living in every habitat type, really. So snails do not have a backbone. They're part of our invertebrate collections that are animals without backbones. They also don't have an exoskeleton, but that doesn't mean that they don't have some kind of protection. They have their shells, which are able to protect them against predation and drying out because they're soft-bodied animals. So the shell in the snail is starts out as a tiny kind of mini snail called a protoconch. When the animal starts to grow, it expands by laying on calcium carbonate, which is shell material, in concentric rows, eventually making a spiral. Sometimes the protoconch will dissolve, and sometimes it's just a tiny little mini shell on the top that you can just barely see. So this shows the arrangement of the spirals, and the animal is actually inside most of these spirals. But this cross-section allows you to see how it's arranged here. And some snails actually have these proteinaceous opercula, which is like a trap door that the snail can use to pull its soft body inside, and they're a little bit mobile inside of their shell. They can't come out of their shell, but they can move in and out of their shell slightly. So they can retract their bodies inside of their shell and close off this little trap door operculum here. Here's another example of a cat's eye operculum in another tropical shell. I mentioned we have a large collection of land snails in our collection. The land snails tend to have really more thin and fragile shells, maybe with the exception of some of the larger species. So we have, this is a giant African land snail. This is the largest of the two species that I brought out here. This species is native to West Africa and other areas of Africa. This snail is actually, they're both called the giant African land snail, but they're different species, which is the problem with using common names, which is why scientists always use species names, genus and species. But these are fairly good-sized snails that have actually invaded in areas that they're not native. And as far as I know, this species of snail, this species of giant African land snail, are present now on every continent except for Antarctica. So they get around, and they cause a lot of damage to crops. They cause a lot of damage to gardens, as you might imagine, and they can also outcompete the native fauna of that area too. So if there's a snail kind of hanging out doing its thing, another snail from another part of the world comes, that snail isn't coming with its predators, so it kind of has this predator release, and it can just go wild and start breeding and start just proliferating. So the problem with invasive species is a big deal. We have a lot of really historically important land snails here. The Academy has a really long history of working in the Galapagos Islands, and we have a very large historical collection of Galapagos land snails, and many of these land snails are either critically endangered or otherwise endangered or vulnerable, or we believe them to be extinct. And these animals are actually still being studied, so we don't really understand the differences between all of the species. Not all the species have been described in this group. So it's really important that these museum collections exist, because scientists that are studying these groups can actually reference museum materials for species that have long gone extinct. Interestingly, in the Galapagos Islands, if you've heard of Darwin's finches or the tortoises of the Galapagos Islands, so there are it's called adaptive radiation, where the animal will adapt its morphological features over evolutionary time to fit its little niche, its little habitat, its piece of the pie, how it eats, how it lives. And then in the Galapagos finches, the beaks actually will be morphologically different depending on what it's eating and where it's living, how it's making a living. So Darwin's finches are a great and the quintessential adaptive radiation example. However, the poorly known example, which is a much cooler example in my opinion, because there are about 70 to 80 species of Nasiotis Galapagos land snails endemic to the Galapagos. So all of these factors, elevation, humidity, predation, other aspects I'm sure of their environment, all contribute to a really wide variety of these snails in their shape and in their the thickness of their shells. So the up at the higher elevations, they can be darker and they can have a wide aperture like these snails do. They can be kind of rounded. They can have kind of a thinner shell, and which is less expensive for them to produce. They don't have to use as much calcium carbonate because they don't have to worry about the desiccation. And that's with the thinner shell, they don't have to worry about the desiccation. The predation, though, they might have to worry about. If they have a strong shell and the bird can't get through it, then that's better for them. So when they're living in a less humid environment, like down by the coast, not at high elevation, they should be lighter because they are more protected against desiccation. They probably will have a bit of a thicker shell because, again, desiccation, but they need to make sure that they're matching their background because of predation. So some snails will actually become more narrowed and have a really small aperture, like these are the same genus, different species, endemic to the Galapagos. So you can barely see the tiny aperture here. The aperture is the opening of the snail where the body comes out, and that's to prevent desiccation. Whereas these snails have a large opening, and you can imagine that a lot of water's coming out of them just into the air. So moving on from the Galapagos, we also have these beautiful Cuban painted land snails. And these guys are not painted. These are actual, the color of these snails. Most are tree dwelling. The species is tree dwelling. And they're very beautiful in color, and the color is polymorphic. So the same species can have all of these colors. These are all the same species. Moving on from the land snails to the marine snails, this is the world's largest snail species that's still living. There may have been some fossil shells that are larger, but this is a syrinx. So they get to be about 3 feet long and weigh about 40 pounds. This one's actually pretty heavy without the tissue inside of it. So they can be pretty heavy. I'm going to put it down because it actually is really heavy. It's the Australian trumpet shell, and they live on sand flats in Papua New Guinea and Australia and that part of the world. And they cruise around. This one's upside down, so they cruise around like that along the mud flat, and they have this long siphonal canal that will hold a 10-inch long proboscis, which is sort of an eating device for the snail. And they'll use their proboscis to suck really long polychaete worms out of their tubes. So some of the worms that they eat are up to 1 meter long, and they're burrowed in the mud in a tube. So they cruise around, suck up a few worms here and there, and then just keep on cruising. Okay. So marine snails actually have much more biomineralization than the land snails. So we mentioned the land snails tend to have really thin shells, whereas the marine snails have much thicker calcium carbonate shells, most of them. Some of them aren't quite that thick, but they do tend to build structures. Like here is a Venus's comb murex, and all of these spines are purposefully built for the animal. The theory is that these spines may prevent the animal from being eaten. I mean, getting a mouthful of spines if you're a fish probably isn't that enjoyable, so maybe the fish decides to go on to something that's a little bit more delicious looking. Also, it might help to not have the animal flip over if there's a high surge area, and it may keep it, if it was in soft sediment, it may keep it kind of from sinking down into the sediment. So there's lots of purposes that spines can serve. Here's another spined turban. This is a star turban, and these are really beautiful snails as well. So one of the things that is incredibly important when we decide whether we're going to take collections or not is whether the collections come with data. The data is actually more important than the specimen to us, because these collections are really a library of life, and they allow scientists and lay people to understand the natural world and to document changes in the natural world. So this is a cone snail, and cone snails are really interesting animals. These are predaceous snails. They're predators that cruise around and look for food. They have a long proboscis, but they also have a harpoon. And their harpoon actually has venom glands that's able to, they can release their venom into the water to cause their prey to be anesthetized, but they also force their harpoon-like tooth out and puncture their prey, injecting venom into their prey to immobilize their prey so they can suck their prey down into their gullet. This particular snail is a cone snail. It's Conus geographus, and this is a fish-eating snail. So this guy, here's a larger one. This guy can actually eat small fish. So there are many, many species of cone snails. I believe hundreds of species of cone snails, if I'm not mistaken. Scientists are still trying to understand the boundaries between species. And so we do have a lot of certain species, and we acquired them from the 1830s on to present. So they're not all collected from one place, obviously, or by one person. So having these comparative collections is really important. So this is one of maybe 2 or 3 drawers that we have of this one species, Conus textili, which is another predaceous cone snail. And this is supposed to be all one species, but if you look at the differences in the patterns in all of these, the conditions, the dimensions, some of them are narrow, some of them are thick, some of them are just darker and lighter, some are not in great condition. You can see that there's a lot of variation within a species. And so it's really important to document that. This might be more than one species, but we need an expert in this group of cone snails to determine that. These are cowries. And cowries are also very beautiful animals. Cowries are very different from the other shells that we've looked at in that their aperture is long and narrow. And they're as shiny as they are, not because they've been polished, but because the mantle of the animal, which is the thin layer that secretes the shell and occurs between the body organs and the shell itself, is able to kind of move against the shell over time, and it creates this really smooth look as if it's been polished. It's kind of been polished by the mantle. So last but not least, I was going to show you a really interesting type of shell. So we've seen the shells that actually have built their own spines through time as they lay down their calcium carbonate chemicals. But this animal, instead of building a shell, actually wears jewelry on itself. It actually uses the same secretions, but some of the secretions are more like a glue in this animal. This is a carrier shell. And the animal, if I turn it upside down, this one is not quite as fragile, which is why I picked it, but if I turn it upside down, you can see the snail. So the snail starts out as just a cone-shaped, innocuous-looking white snail. But on the top of the snail, it starts to glue rocks and shells and objects, sometimes bottle caps or different debris around itself. And they can be very intricate. This one is mostly shells and rocks. This one, let's see if we can see this one a little better. They're pretty amazing shells. So this one has barnacles glued to the top of its whorls here. So they actually, the animal will clean off the area that it's going to glue something onto them, and then manipulate using their foot, which is the structure that's in their operculum that they cruise around on. I didn't mention that snails use their foot to glide around on a surface using a mucus that they secrete so that they can more smoothly glide and not injure themselves on a rough surface. So they'll use that foot and their tentacles and their proboscis, and they'll maneuver the object into exactly where they want to glue it, and set the glue to that area, attach the object, and then they have to wait for a period of many hours for it to harden and solidify. So why do they do this? Well, they don't know for sure, but what we think is that most of these live in fairly deep water. They can live in softer sediments like muddy sand. So it could be that it prevents them, it makes kind of a little cage and prevents them from sinking into the mud. It also doesn't let them be flipped over quite as much. but more likely it prevents against predation. So it does create kind of a little protective cage from the soft parts, for the soft parts here. So if a fish comes along, they're less likely to want to take a bite out of this or a bite out of that than they are to take a bite out of a nice smooth snail that they know what they can get their mouth on it and manipulate it. This one's going to be really difficult to manipulate. Also, the structure of the objects that have been glued to it also are somewhat protected too. This one's picked some pretty thick shells and barnacles, which are hard to get to. Some of them actually put sponges on their shells, and they can be really beautiful. This is a smaller one, but it's out of the bag, so I thought we could see this one. Here you can see where it looks like one of the shells has fallen off over time, and this is where it was glued, this little space here. So you can see how they cleaned the area and then put in the glue, the mucus glue, and then attached it to these little circles here. So over time, they've probably fallen off before it was donated. So this is a sponge. It looks like a glass sponge, and this is a pretty common type of sponge to be adhered to some of these carrier shells. They're in the genus Xenophora. This one, to me, is pretty amazing. So I'll carry it over so you can see it, but it's incredibly fragile, so we have to be careful with it. So this is, I don't know if you can see the shell. The shell is down there, and it's a tiny shell. It's about this big. So there's a shell there, and it's added various snails and one smaller sponge here, but then it decided it was just going to go for it, and it got this huge sponge and attached that. Well, thanks so much for looking at some of our incredibly diverse and interesting shell collections. You only saw a very small portion of our massive shell collections here in the Department of Invertebrate Zoology and Geology. And I thank you all, and enjoy seeing the snails out in your garden.
Hi.
Hi.
I love the crafty snail at the end there. It's amazing.
It wears hats.
It wears hats.
So good.
Yeah. Thank you so much to Mandë and Jann and Chrissy for this amazing night school. Chrissy is actually, the reason we pre-recorded is she's actually in the field in Roatan with our Hope for Reefs team. So maybe she'll have some more cool stuff to share when she gets back.
Yes, indeed. And we here at Night School come to you live on the first and third Thursdays of each month. And our next program, so we talked a little bit about snail shell aperture, but our next one is going to talk about aperture with photography. Haha. It's a program called Nature in Focus. It's going to be on July 7th. And we're going to bring on some amazing photographers and storytellers who are dedicated to documenting and sharing the wonders of our natural world. So stay tuned.
And also we saw, Jessica, your question about book recommendations. So we sent a quick message to Mandë and Jann. And yeah, that was Mandy's recommendation. I put it in the chat. It's Privileged Hands: A Scientific Life by, I'm gonna mess up the same thing, Curator Twila. But yeah, it's in the chat. Yeah, so again, yeah, we're coming back to you on July 7th. And yeah, subscribe to our YouTube channel so you get notified and you won't miss anything. And if you want to share this night school with your friends who also love snails or you think have high potential for liking snails, this stays on our YouTube at the same URL. So thank you so much for being with us tonight. And yeah, have a good night, everyone.
Take care, everyone. Good night.